A very warm welcome everyone to this latest in our regular series of Precision Nanosystems Tea Time webinars. It's our great pleasure this afternoon to welcome Dr. Enrica Chiesa of the University of Pavia, and she will be talking about hyaluronic acid-based nanoparticles for selective inhibition of CD44 positive human mesenchymal cell proliferation. So just before we get started, I just have a little bit of housekeeping to do. So firstly, just to introduce the Europe team. So my name is Richard. I act as field application scientist for the north of Europe in Benelux, Scandinavia, the UK and Ireland. Also joining me on this call and running the question and answer session will be my colleague Martin Rabel, based in Germany, and he looks after Central and Southern Europe. And we are also helped and supported by our sales team. We have in the north of Europe AJ Johnny, who's the regional sales director for the north of Europe, and Central and South Europe is Jürgen Schmidt-Söchtig. And with that, it's my pleasure now to hand over to Enrica, and over to you, Enrica, and everyone, I hope you enjoy the webinar. Thank you.
Okay. Good afternoon to everyone, and today it's a pleasure for me presenting our last work on the microfluidic technology for the synthesis of hyaluronic acid-based nanoparticle for the selective delivery of drug into CD44 positive cells. Our idea is to target CD44. CD44 is a transmembrane glycoprotein widely distributed in normal adult healthy tissue, and it is composed by an extracellular domain with the ligand binding site, a variable region whose variation leads to different isoform of CD44, and an intracellular domain liable of the intracellular signaling pathway. CD44 is involved in multiple signaling pathway, contributing to the cell differentiation, proliferation, and migration. CD44 is a molecular marker of mesenchymal stem cells, and it is involved in mesenchymal stem cells requirement during tissue regeneration and development. Otherwise, CD44 has been correlated to different physiopathology. It is a marker for cancer cells, and its overexpression is confirmed in different types of solid tumor. In this case, CD44 is involved in cancer cell proliferation and metastasis. For all these reasons, research efforts have been focused on studying the prognostic and diagnostic value of CD44 and how to exploit it as a target agent. Hyaluronic acid is its main natural ligand, and it can be used as anchor of attach to CD44 to improve nanoparticle selectivity against CD44 positive cells. Hyaluronic acid is already used in biomedical fields since it is a biocompatible, biodegradable, and non-immunogenic polysaccharide. Our first attempt to exploit CD44 for a selective delivery of drug into cells is reported in this paper of 2018. The rationale was the selective delivery of EVEROLImus in lung allograft-derived mesenchymal cells. EVEROLImus has been successfully used in combination with corticosteroids and cyclosporine after lung transplant for the prevention of bronchiolite obliterans syndrome, which is a fibrous obliteration of small airways due to abnormal mesenchymal cells proliferation and extracellular matrix deposition, causing bronchiolar occlusion and organ rejection. First, we set up conventional bulk mixing method by adding dropwise a chitosan solution at constant flow rate with a proper needle into a mixture of hyaluronic acid and TPP. The system was under magnetic steering, and the magnetic steering was maintained to allow the nanoparticle stabilization. The optimized preparation protocol allowed to obtain a nanoparticle with a size below 200 nanometer, good polydispersity index, and negative zeta potential, ensuring the hyaluronic acid deposition onto the nanoparticle surface. These nanoparticles were tested in vitro in mesenchymal stem cells recovered by bronchoalveolar lavage from bronchiolite obliterans syndrome patients. We test the cell viability and the nanoparticle uptake profile. The positive results in terms of biological performances were associated with some limitation correlated to the nanoparticle synthesis. In particular, we have found a lack of batch-to-batch reproducibility and an important operator variability. So we decided to move towards microfluidic. Microfluidic was born to mitigate this concern, and the miniaturization allowed two properties not otherwise possible at macro scale. It is characterized by outstanding reproducibility. A computer control removes the batch-to-batch and the user variability. It has an intuitive flow work. We are able to tune the particle size thanks to the precise control over the fluid flow rates. It is a versatile technique. We can formulate different types of drug into several nanoparticles, lipid and polymer based. It is speedy and efficient, and it is easy scalable. Our work seeks to develop a one-step microfluidic technique for the synthesis of hyaluronic acid chitosan nanoparticle by electrostatic complexation. Not many papers have demonstrated the feasibility of nanoparticle synthesis by microfluidic by electrostatic complexation. And even if the microfluidic benefits are well known, new synthesis procedures need to be investigated. The optimization was aimed towards obtain a nanoparticle with customized features with a potentially strong active targeting to CD44 positive cells. For the nanoparticle preparation, we use the nanoassembler batchtop platform. The chitosan solution and the mixture of hyaluronic acid TPP solution were injected into separate chamber of the microfluidic chip, and the pattern into the grooves caused the repeated and the rapid folding of the two solutions, leading to increase the surface area at the interface and reducing the diffusional distance, leading to an increase of the mixing rate. After a preliminary experiment that permit to identify the concentration range of each compound, hyaluronic acid concentration was varied from 0.4 to 0.15 milligram per milliliters. Instead, screened chitosan TPP weight ratio were 50, 15, 25, and 12.5 to 1. Nanoparticles were then characterized in terms of mean size, polydispersity index, and surface charge. Statistically analyzed results revealed the important role of hyaluronic acid concentration and the ratio between hyaluronic acid and chitosan on the nanoparticle features. In particular, at first instance, hyaluronic acid concentration increase triggers an important nanoparticle enlargement, while the ratio between hyaluronic acid and chitosan is label of the nanoparticle surface charge. From TEM images, nanoparticles mostly appear like spheres, and the identification of both hyaluronic acid and chitosan components and their interaction during the polyelectrolyte complex formation was evaluated by FTIR spectroscopy, and the FTIR spectrum of nanoparticle was compared to those of the raw polymers, and nanoparticle spectrum presented several characteristic vibration of hyaluronic acid and chitosan, but they are intensified and shifted to high wave number. The signal displacement confirm the presence of both macromolecules into the nanoparticle. Moreover, a new shoulder appears here, and it is reported in literature that is an evidence of the hyaluronic acid protonation occurring during the polyelectrolyte complex formation. The uptake profile of our nanoparticle was tested at 30, 90 minutes, and 4 hours of incubation. Here I reported the confocal images at several time points, and as you can see here, after 30 minutes of incubation, nanoparticles are widely distributed into the intracellular compartment. The nanoparticle signal is more evident at 90 minutes of incubation, and finally, after 4 hours of incubation, there is a notably high concentration of nanoparticle inside the cells. The internalization of our nanoparticle is better revealed from the 3D images obtained by the confocal images elaboration along the z-axis. And as you can see here, we have on the top the cell membrane where CD44 is stained in green. And here inside we have the nucleus and the red nanoparticle widely distributed into the cytosol. Quantitative analysis permit to claim that the red fluorescence inside the cells arise from 30 minutes to 4 hours of incubation, regardless the concentration of nanoparticle used. From the 3D images, also some cell membrane protrusion were observed, leading us to think about endocytic vesicle, and it encouraged us to deeper investigate these entities, and the presence of endocytic vesicles was confirmed in this cartoon, reporting the confocal images of human mesenchymal stem cells after 90 minutes of incubation with the nanoparticle. Both images present endocytic vesicles, and then the red fluorescence and green fluorescence intensity along the yellow line across the selected vesicles were determined. And as you can see here, the peak of the red fluorescence of nanoparticle is in the middle of the peaks of the green fluorescence of the cell membrane. So we can claim that our nanoparticles are located into a discrete cytoplasmatic vesicle-like region of approximately 1-3 micrometer in diameter near the cellular membrane. To understand if our nanoparticle could have access to the cells thanks to the specific interaction with, between hyaluronic acid and CD44, competitive binding experiments were carried out. The rationale of this experiment consists in comparing the uptake profile of hyaluronic acid-based nanoparticle in mesenchymal stem cells cultured in normal condition with the uptake profile of hyaluronic acid-based nanoparticle in cells pretreated with a specific anti-CD44 primary antibody. Pretreating with anti-CD44, the interaction between hyaluronic acid and CD44 should be prevented, avoiding the nanoparticle uptake. For further control, plain chitosan nanoparticles were used. In this case, pretreating with anti-CD44 primary antibody, the nanoparticle uptake should not be affected. As you can see here, pretreating with anti-CD44 primary antibody, the red fluorescence of hyaluronic acid-based nanoparticle inside the cells is almost negligible. Instead, chitosan nanoparticles are massively internalized into the cells, and these quantitative results are confirmed by the confocal images. Finally, EVErolimus loaded nanoparticles were prepared by adding EVErolimus ethanolic solution at different concentration. Two different chitosan EVErolimus weight ratio were tested, 2:1 and 5:1. The best results in terms of encapsulation efficiency was obtained with the drug ratio 5:1 with encapsulation efficiency of around 50%. Instead, the physical property of our nanoparticle are superimposable to the placebo contour part. TEM images confirm the spherical shape of the nanoparticle, and on batch 2.4 and 2.7, in vitro release test was performed. And as you can see here, both formulations are able to prolong the drug release till 16 and 48 hours, compared to the drug dissolution that is completed in 2 hours. Finally, we tested the effect of EVErolimus loaded nanoparticle on human mesenchymal stem cells. First, the cytotoxicity by MTT assay, and as you can see here, the placebo nanoparticle did not show any toxic effect, confirming the well-known biocompatibility of the raw polymers. Instead, EVErolimus solution is 5 times more toxic than the nanoparticle contour parts. The effect of EVErolimus and our formulation on the mesenchymal stem cells proliferation was then tested by tracking the DNA synthesis by bromodesoxyuridine assay. Why is important this test? To understand the importance of this test, I need to give you a brief background about the EVErolimus mechanism of action. EVErolimus inhibits mTOR by preventing the binding of receptor to mTOR1, leading to a downregulation of cyclin D and cyclin E, and the cellular events mediated by EVErolimus are the cell cycle arrest in phase G1, growth retardation, and then apoptosis. As you can see here, after 30 minutes of treatment, the highest concentration of EVErolimus loaded nanoparticle are able to inhibit the cell proliferation more than the placebo contour part. Moreover, the mesenchymal stem cells proliferation inhibition is dose dependent and increase with the increasing nanoparticle concentration. Finally, after 4 hours of incubation, no proliferative cells were detected at the highest nanoparticle concentration, and the inhibition of the cell proliferation was more effective than the EVErolimus solution. So in conclusion, microfluidics assisted ionotropic gelation method can precisely induce the electrostatic interaction between hyaluronic acid and chitosan to produce nanoparticle with tailor-made size, loading EVErolimus in output manner. Nanoparticles are able to cross the cell membrane and to reduce the proliferative activity of CD44 positive cells. Both chitosan and hyaluronic based nanoparticles are able to be internalized, but we have demonstrated the different mode of internalization ascribable to the different binding form to the cell membrane, through CD44 for hyaluronic acid-based nanoparticle and through a specific electrostatic interaction for the chitosan nanoparticle. So finally, I want to thank all my collaborators, my supervisor, Professor Ida Genta, and Antonietta Greco, who is the PhD student that works with me in this project, and all the collaborators. And also thank you for your attention.
Yeah, thank you, Enrica, for this excellent talk. It was really a pleasure to having you here. And we're on the top of the hour, so it's time to say goodbye to everyone. Thank you, Enrica, for being here. Thank everyone for... Yeah, we thank everyone for listening to our Tea Time webinar, and we hope to see you soon in 2 weeks for our next one. All right. Bye-bye. Have a good evening. Have a good afternoon, everyone.